So now we're going to compare unicellular and multicellular organisms. So unicellular organisms are going to be slightly different than multicellular organisms because although they both perform a variety of activities because they both need to live, multicellular organisms we can compare to humans. And cells are going to be organized into tissues and organ systems. And we know that tissues and organ systems perform life activities. However, in unicellular organisms, like a paramecium, organelles perform all of life's activities. So organelles are known as tiny organs, and they perform all of life activities for unicellular organisms. For multicellular organisms, we, like humans, we have tissues that make up organ systems, so much larger than organelles. But our organ systems are made up of tissues, and those tissues are made up of cells, and those cells are made up of organelles. Now, how do we compare a unicellular organism like a paramecium to a multicellular organism like a human? So, life activities, we have separates, protects organism from environment. So that means your skin for humans. For a paramecium, that would be their cell membrane. A life activity, another one, would be movement from place to place. We, humans, multicellular organisms, have muscles and bones. Paramecium only have cilia to move. And what coordinates and controls activity? So for multicellular organisms, that's going to be our brain. For a paramecium, they only have their nucleus to control and coordinate their activities. What about excretion or removal of waste? So humans have an excretory system, and parameciums have a cell membrane. Gas exchange. Well, we have lungs that control the oxygen we breathe in and the carbon dioxide we breathe out. Paramecium, again, their cell membrane. Remember, unicellular organisms do not have organs. They have organelles, so they only have those very small organs to make them up. Break down food and nutrients. We have a digestive system that's made up of many different organs. For a paramecium, the unicellular organism, they have a lysosome vacuole. Remember, lyse means to break, so a lysosome vacuole will break down food and nutrients. So now we're going to look at the different techniques of studying cells. The first one we're going to go over is called microscopes. And microscopes, there's different types. So the first one we're going to go over is called a light microscope. Now, in most school settings, you see a compound light microscope. So it's the main microscope that we use in class. It uses two lenses to magnify the image. So you could think of two magnifying glasses, one far away and one close to your eye. Light must pass up through the objective, which is a long tube, so that we can actually see the very small specimens underneath of the objective lens. So specimens have to be very small and thin to be viewed. Larger specimens can be sliced into thin sections to be viewed, and specimens, really neat, can be viewed alive. So if we were to take pond water and place it onto a slide and look at it underneath of a compound light microscope, we would be able to see very small organisms moving. That's something very important about the compound light microscope. Now, we can also use stains in order to make different structures, like a nucleus of a cell, stand out. However, keep in mind that stains will usually kill the specimen, but as a scientist, sometimes we need to be able to see different images within our slides. And here are some examples. So we have bacteria, we have different types of cells where we can actually see the interior of that organism. The next microscope, a stereoscope or dissecting microscope. So this is used to observe 3D structures of specimens. Pretty low magnification, and it magnifies only up to 50 times. Now, I know that sounds like a lot, but we cannot see cells. This is a type of light microscope, very easy to use. It has a single lens. The light source is directly above the specimen compared to our compound light microscope. It's actually underneath of our specimen. And for this microscope, the specimen does not have to be thin. And here are some examples, but we can see the exterior of a leaf. We can see bugs, but we cannot see inside of those cells. So we can't see the cells. Then we have an electron microscope. This microscope can magnify up to 1 million times. The specimen must be dead and coated in metal. This is a very expensive microscope and it's not easy to use. Then there's the scanning electron microscope. We can see a 3D image. It looks at the surface of the organism. So just like we see in the picture to the right, we can see the surface. But we cannot use this microscope to look at living organisms. So we can see the surface of red blood cells, but we cannot see the cells inside. Here we have an organism. We can see again the outside, the exterior. Once again, we can see the exterior of a flea. Pollen, but remember, we cannot see the cells that make up these different organisms. Here's the lining of our lungs, but remember, a compound light microscope, we would take a thin slice of the lining and be able to see the cells. With a scanning electron microscope, we can only see the surface of the lining. Here's what a light bulb filament looks like. And finally, our last microscope we are going to go over, a transmission electron microscope, also known as TEM. So this is a 2D image. So on the right, you see the difference between a mitochondria and a chloroplast. We can look at the relationship between the internal structures. And again, this microscope cannot be used to look at living organisms. So what happens with staining? So staining helps us look at cell parts by making them more visible under a microscope. So we could use iodine for a plant. Basically what iodine will do is it will stain the sliver of cell that we have sliced, and we will be able to highlight specific features within that cell or cell parts. Methylene blue is used for animal cells, and again, methylene blue will help us highlight specific cell parts to make them more visible underneath of a microscope.